So it's my pleasure today to introduce Professor Gerard Ateshian from Columbia University. After sort of wandering the world for many years, from Lebanon, spent some time in London, landed, I guess, in the U.S. in Boston for a little bit, and eventually then in New York City at Columbia. Right. Started his bachelor's work there, and did his bachelor's through his Ph.D. At Columbia. You know, people oftentimes give me a hard time because I did my bachelor's through postdoc at the University of Michigan. And I would say, well, I had great football tickets there. So, I don't know what his excuse is. He said there's really good fencing there or something like that. Better school. Better school, there we go. All right, yeah. And much better than Penn, both of them. So, but he took it one step further and actually joined the faculty in 1991 at Columbia, and 20 years later is still there.
That's right.
Professor of mechanical and biomedical engineering at Columbia. Professor Ateshian has worked in the area of looking at fundamental biomechanical issues associated with cartilage and joint mechanics. Has published over 150 papers and book chapters, primarily in that area. Really improved our understanding. And then I think it was about 5 years ago you started dabbling in the area of tissue engineering a little bit and looking at sort of the role of mechanics and mechanical environment on the development of constructs for treating injured cartilage. And I think that's one of the things you can address today. So thank you very much for being here.
Thanks, Bob. Thank you. I'm happy to be here. Bob, we were just having dinner yesterday, and this morning I was talking to Cheng about how we lose track of time. I actually started doing tissue engineering about 11 years ago, but but it's true. I gave a seminar here a few years ago, but I don't remember how many years ago. So if there are graduate students here who saw me the first time, I'm really sorry that you're still around. But in any case, so I will focus. The first seminar that I gave was more on cartilage lubrication. I'm going to touch upon that briefly today to explain the structural function relationships in articular cartilage. Let me show you what cartilage is, so we'll understand, what we're talking about. Do we want to turn down the lights? Okay. So again, I'm going to talk to this audience without necessarily assuming that you know much about articular cartilage. This is your knee joint. This is your kneecap. This is the femur or thigh bone. This is the tibia or shin bone. And the white layer that you see here is articular cartilage. So this is a cross section from MRI, and this is what a healthy joint looks like. And you can see that cartilage is very smooth, white, glistening. The patella, which is up here, has been reflected down. That's what you're looking at. So it's been cut and moved down. So you see that it's embedded, in fact, in your quadriceps tendon that then inserts into the tibia. This is what happens to cartilage when you have osteoarthritis. It wears down all the way to the bone. You can see actually exposed bone here. And this is the patella as well. So articular cartilage has its main function is to transmit loads across your joint. So you can do all these kinds of motions, and it has to give you low friction and wear. That's its main function. It achieves this by having the following composition and structure. It's mostly water. Cartilage is mostly water, and the water content changes through the depth. This is a cross-section of cartilage. This is the articular surface. This is the subchondral bone to which it attaches. This is the collagen in articular cartilage. Is type 2 collagen. This is an AFM of the surface of cartilage, and there is about 20% collagen in articular cartilage. The remaining major dominant constituent of articular cartilage is proteoglycans, which consist of about 3 to 10% by wet weight. And so the collagen gives the tissue tensile properties. The proteoglycan contributes to the compressive properties. They're enmeshed in this manner. And so if we're interested in engineering cartilage, the dominant constituents that we need to worry about are collagen and proteoglycans. There are also other issues that we'll need to worry about. So here's actually a movie, without the sound, of articular cartilage. This is exactly the same joint we saw a moment ago, although it's oriented in the opposite direction. This is the patella. It's an actual cross-section. So we're looking at half a knee joint. This is the femur, and the black speckles that you see were sprayed on to create a pattern that we could then use to measure strain. So we're applying one body weight on half a joint, which is 2 body weights on a full joint, and you can see how much deformation the tissue is undergoing. Though cartilage is known as a soft tissue, it is nothing as soft as, say, tendon or muscle when it's not, activated. So therefore, it's actually a fairly stiff tissue, and the deformation that it undergoes is relatively small. The strain patterns that you will see from these types of analyses are shown here. What you have is that the legend shows you the range of strains that we have is -7.5% to 8.5%. This is just one representative joint. So when you apply about 2 body weights across the patellofemoral joint, you'll only get compressive and tensile strains that range from roughly -8 to 8%. So there's very little strain in these tissues. It's a very resilient tissue. What makes cartilage, so stiff under physiological conditions? Well, it turns out that the interstitial water of cartilage plays a critical role. It is, after all, about 80% water, 90% near the articular surface. If you take a piece of cartilage and you load it in a confining chamber and you measure the fluid pressure on one end and the total load on the other end, what you find is that the fluid pressure, which is shown with the experimental data, the orange dots down here, starts out very high. It's actually equal to the stress that you apply. And then over time, as fluid escapes the tissue, the pressure will go down. So in this confined compression configuration, where this sample is not allowed to expand, 100% of the load is supported initially by the fluid, which is a very important thing to keep in mind. What else characterizes cartilage? Well, it is a collagenous matrix. So when you actually apply tension on it, this is a stress-strain curve. On the vertical axis is the stress. On the horizontal axis is the strain. And you actually have a range of compressive strains here, which are negative, and tensile strains, which are positive. You see the slope of the stress-strain curve is much higher in tension than in compression. It could be up to 100 to 1 difference or ratio between the tensile and compressive modulus of cartilage. That comes from the collagen. What is the functional role of this tensile modulus? After all, isn't cartilage loaded in compression? Why in the world would you care to have such high tensile stiffness for articular cartilage? The answer came from theory. We found out from theory that if you take into account this tension-compression nonlinearity, and if you look at configurations other than confined compression, if you load the tissue in unconfined compression where it wants to expand laterally when you compress it, the very high tensile stiffness will prevent it from expanding laterally. Therefore, the fluid will pressurize more to counter the compressive load. And therefore, whereas theory predicts a much lower fluid load support if you have the same modulus in tension and compression, if you have a different modulus, a much stiffer modulus in tension, you will predict a very high fluid load support approaching 100%, even for configurations other than confined compression. And these are experimental results that confirm this. Again, the time dependence is because if you load the joint statically, which you can do in the lab, then eventually the fluid pressure will subside. So what is the functional role of the fluid pressurization? It makes the tissue stiffer, so you don't compress the tissue too much, therefore you don't kill the cells every time you step on your joints, or on your anytime you walk. And so what happens is, that stiffness is very important. Another thing that cartilage does is give you very low friction. The friction coefficient is the ratio of the frictional force over the normal force. And we found that if you actually model the fluid load support as this ratio here, you will find that the friction coefficient will be smallest when the fluid pressure is highest. That was a hypothesized model. I spent the first 10 years of my career on this. It's only one slide here. I'll just show you the result and we'll move on to the things I did in the second 10 years of my career. So here it is. The fluid load support is very high. You're applying a static load and you're measuring the friction coefficient, a piece of glass sliding on a cylindrical piece of cartilage. You find that the fluid load support is close to unity, or nearly 100%. After about 20,000 seconds, it has subsided. The friction coefficient is initially very, very low when the fluid pressure is high. How low? The answer is down here. It's 0.004. For every 1,000 pounds of compressive load, there's only 4 pounds of frictional force. That is exceptionally low. There's like no, no bearing that's man-made. Your car engine, your oil, lubricated bearings give you friction coefficients on the order of 0.01, 0.02, not 0.004. So it's quite remarkable that we get such a low friction coefficient. But if you let the fluid load support subside, then the friction coefficient will rise to something really bad, something as high as 0.28. That's a very bad friction coefficient. We spend quite a lot of time showing that this will never happen in the body if your cartilage is healthy, so you don't have to worry about it. It only happens in the lab when you can statically load for about 20,000 seconds. Anyway, so that gives you a sense of the functional design, the structure-function relationship in cartilage. So if we are trying to grow cartilage for the purpose of tissue engineering, and if we want the tissue to be functional before we put it in the body, then we have to be able to reproduce these kind of properties in vitro before we implant it. That is our philosophy. When I say ours, Clark Hung and I have been collaborating over the last 15 years in this field, and so other people in the field think that the body should be the bioreactor. You should put the right environment, the right scaffold, the cells in the body, let the body grow the tissue to its functional level, and that's certainly a valid alternative. But we feel that the environment of cartilage is so harsh, the loading environment is so harsh, that it's unlikely that a fledgling construct will survive unless it starts out with very strong properties. Now, I've raised your expectations. Don't expect me to show you working cartilage at the end of this lecture. If it did, I wouldn't be here. I would be in some private island enjoying myself in the sun. Okay, so what did we do? The first thing that we discovered is that, so based actually on Bob Nerum's work, who had done the work actually in vascular tissue engineering, what we wanted to do was to see if mechanical loading of chondrocyte-seeded agarose gels, which is what you see here, would enhance the properties of the tissue. So no one had done that in cartilage before we started that. We created a very simple bioreactor. That was my introduction to the fancy word bioreactor. When you see this, you think to yourself, wow, that's all it is. It's a petri dish, loading device, a cam that rotates continuously, so this will go up and down by a small amount. This is what we found if we didn't load the tissue. This study was done in 2000. So this is what the gel looks like at day 0. At day 28, it's starting to look whitish, a little bit like cartilage. The compressive stiffness is about 20 kilopascal. For bovine cartilage, the compressive stiffness normally is about 500 kilopascal. So that shows you how far we are from that result. With dynamic loading, we go up to 100 kilopascal, and you can see that the difference is quite remarkable. So clearly, dynamic loading enhances the outcome. What is very interesting, and I don't have time to show all the results, but the biochemical composition for the two major constituents, which is collagen and proteoglycans, is the same in the two groups. You actually are not producing more matrix, so there's this question of what is the dynamic loading doing, and I'll give you some ideas as we go along. Anyway, we got really ambitious and we said, great. One of the challenges of tissue engineering is if you have these little cylindrical constructs and you put them in defects, well, you saw that image of arthritic cartilage a moment ago. There are no nice circular holes for your tissue constructs, so you got to actually look at larger patches, and then you have to worry about integrating these larger patches of engineered tissue into these defects. So we figured, in the long term, most likely when you actually want to replace the cartilage of an arthritic patient, you might as well consider the possibility of doing the entire articular layer. I mean, why stop at 5 millimeter diameter cylinders or 10 millimeter diameter? Might as well think big. And we thought very big, and so we used, this is once again a human-sized patella. Would you believe it? This is actually, this is the scale bar. This isn't a small scale version. We were able to culture these very large constructs in these urine sample cups, and we cultured them for a few weeks, and this is what happened. And we used trabecular bone so that with the idea that if we poured the agarose gel directly in it, eventually it would anchor very nicely. This was a proof of concept for us. We did a number of things, creating the molds for reproducing the shape using data from MRI, cutting the bone using CNC machining. There were a lot of things that we had to figure out. The message I want you to take from this is the following. This is what we call a safranin O stain. The darker the red color is, the more proteoglycans you have. What you see is a very good example of how nutrients do not go deep enough into the construct because this white region here has very little proteoglycans. So when you create large constructs, your histology shows very clearly that that the cells which are closer to the surface are using up most of the nutrients before they have a chance to arrive at the center. So the properties of these constructs was very poor. It wasn't even the 100 kilopascal we were achieving with the dynamic loading, but this slide shows very clearly that, you know, the transport of nutrients across a barrier of tissue where cells are using up those nutrients is a major challenge in tissue engineering. So now I'm going to show you results a little bit out of sequence in terms of chronology. What we found out is something that we should have learned early on. One of the things we learned for those of us who do cartilage, you know, mechanics and cartilage tissue engineering, we always tell you cartilage is avascular. It's like there's no vasculature in articular cartilage. That's one of the reasons that cartilage doesn't heal well. In fact, it's only adult cartilage that is avascular. So these slides show you vasculature in immature cartilage. In humans, you will find these kinds of canals. That's what they're officially called until about the age of 16. So it turns out that cartilage is highly vascularized during the time that you need it, when you actually have to grow cartilage. So it seems to make sense that even though people who do cartilage tissue engineering are not familiar with this concept, it seems to make sense that you need to have some form of nutrient supply through, for example, vasculature. Now, Bob has been putting, you know, channels in his bone constructs for a long time, and that's because bone is highly vascularized, and it's not a surprise. And people who do, you know, cardiac tissue engineering, like Gordana Vunyak-Novakovic, who's now at Columbia, also need to put channels in their constructs because the myocardium is highly vascularized. So for us to actually put channels in cartilage was a bit of a leap of faith. When we suggested the idea, surgeons said to us, what, put holes in cartilage? Won't it make it weaker? So here's what we did. We put holes anyway, because we can do that in the lab. It doesn't hurt anyone. So we just extracted a tiny little core, 1 millimeter diameter core, from constructs that were created in the same manner that you saw earlier. We cultured them, and in the usual way, actually not totally the usual way, sometime in 2006 or so, Ben Byers and Rob Mock, who was no longer with us at the time, discovered that if you supply growth factors, specifically TGF-beta 3, for only 14 days, then after you take away the growth factors, then the growth of tissue escalates very rapidly, which is a very interesting point. I'll come back to that a little bit later. So this is what we did, put a channel in, and what we found was significantly better results. First of all, after about 56 days of culture, the channel did start to fill in, which was important. Bob is telling me it's a problem for him in his bone constructs. For us, we want the channels to fill in so we can convince the surgeons that it'll be okay to implant these, constructs in patients in the future. And what you can see is over time, if we look at the properties, this is what happens to the control without channels. After 56 days, the properties plateau. You see here after 14 days, when we release the TGF-beta 3, look at how much the material properties increase. And we are now reaching easily the native properties, compressive properties of articular cartilage, 600 kilopascals. So these properties are wonderful. And if you actually put a channel, you can do almost twice is good, which is remarkable. Now you're reaching 1,000 kilopascals just by putting that one single channel in, which mostly fills in. This is the dynamic modulus, which is not twice as good, but still better. The dynamic modulus depends in part on the collagen content. So far our proteoglycan content reaches native levels, but our collagen content doesn't. Normally what you want this number to be is anywhere between 20 and 80 megapascal, and we're still only at about 4.5. So, but still, you get better results if you put a channel. So once again, what we find is that the GAG content, which represents the proteoglycan content, and the collagen content are grossly similar with or without the channel. So that means that there must be additional quantitatively minor matrix molecules which we haven't yet identified. We have many suspects, but seem to improve the properties. And of course there is a structural reorganization which is probably enhanced. And you can see here the collagen distribution with the channels is actually more uniform than if you don't have the channel. This is the control without the channel, you can see very little collagen at the center, which is again representative of this nutrient limitation. When you put the channel in, it fills in, so there's now collagen in regions that previously didn't have collagen, though you can still see the channel. So it didn't completely fill in, but clearly you get better distribution of the collagen content. And if you actually measure the properties through the thickness, through the depth of the sample using optical methods, and you track the images with digital image correlation, and you get the displacement, and the slope of that gives you the strain, you can calculate the stress and extract from that the modulus. Now look at, this is the construct, the control construct, which didn't have a channel in. So we're looking at the properties from this edge to this edge. So this is what we like to call the M&M effect. Nice and crispy on the outside, soft and chewy on the inside. So this is what happens when you don't have sufficient nutrient supply, and when you actually put the channel in, and we're measuring the properties here, obviously not in the channel, you find much more uniform properties, and also the scale bar is different. So here the average value is about 1,000 kilopascal, here the average value is about 600. So clearly putting the channels in helps. And if you have larger constructs, so we had done a study where we had done 4 millimeter diameter versus 10 millimeter diameter. If you think that the nutrient limitation is mostly through the thickness, then you actually will find out that it's not true. A 10 millimeter diameter construct with the same thickness as a 4 millimeter construct will give you poorer properties. But if you put a few channels here, then you can improve the properties once again. And what you can see here is, the results. Let me guide you through it. So the 4 diameter control is the pink. The 10 diameter with the channel is the yellow. And the 10 diameter without the channels is lower properties than the 4 millimeter. So you clearly get better results with channels in the 10 millimeter construct. There was no channel control for the 4 millimeter in this part of the study. So what we're doing now, we have a grant to optimize the placement of channels in large constructs, and after 5 years of studies, we hope to be able to go back and reconstruct these full-size patellae and show much better properties than that study that we did in 2004. So we're not there yet, that's work in progress. I'll show you more of that. Now let's go back to this study which shows that dynamic loading improves the results. So we asked ourselves, so I'm chronologically going back a little bit. When we found this result, we realized, of course, that dynamic loading could induce the cells to produce more matrix through perhaps integrin-mediated pathways, a direct mechanotransduction pathway where the cells see the deformation and somehow internally decide to produce more matrix, or, it's not necessarily an exclusive or, it could also be that you're enhancing nutrient supply by dynamically loading the construct. So I've shown you convincing data that nutrient supply is critical, even in 4 millimeter diameter constructs. So what can you do with dynamic loading in terms of enhancing nutrient supply? So here's what the literature says. There are 2 types of studies that look either at desorption. So this is, we're looking at the fraction of solutes inside cartilage that's being desorbed with dynamic loading versus static loading as the control. So in the studies published throughout the last few decades, there's been some conclusive evidence that if you dynamically load a construct, you might actually get rid of solutes a little bit faster. That's what this shows. So the fraction of solute desorbed is higher for dynamic loading than static loading. And you can show that with glucose as well as larger molecules such as albumin. Albumin, whose molecular weight is about 60 kilodalton. Glucose is like, just 0.3 or whatever it is. So that's what the literature has said. A study by Larry Benasser showed that if you have a growth factor such as IGF-1, and now we're looking at uptake. The IGF-1 is in the bath. You're going to dynamically load cartilage. He found that after a few hours of loading, you'll find that the uptake of IGF is faster, but eventually it reaches the same value as static conditions. So static conditions means you have passive diffusion. The solutes are simply diffusing in. Over time, the concentration inside is the same as if you dynamically load it. It just takes longer under passive conditions. So we were interested in finding out whether there was something else from theory, from a theoretical perspective, whether we could actually see an effect of dynamic loading. So we started with a theoretical study. And so I've spent a lot of time doing theoretical analyses in biomechanics. Bob referred to that a little bit in his introduction. I'm not going to show you too much theory today, but I'm going to show you the consequences of theoretical predictions. Just like the friction and lubrication was built in great part on theoretical predictions that led to experimental discoveries, it's the same thing that we have here. So we considered a very simple model of a cylindrical disc being loaded dynamically in a bath that contains some solute with a concentration C star outside, and we were looking at the average solute concentration in the disc over time. What we did differently from the prior literature, so let's do a little bit of theory here. Let's understand this is the momentum balance equation, Newton's second law of motion under quasi-static conditions. Inertia forces are negligible enough that what you're saying is sum of forces equal to 0 instead of mass times acceleration, because you consider mass times acceleration to be negligible. What drives the solvent in the mixture is the gradient in its mechano-electrochemical potential. If there's a gradient in the pressure, they will drive the fluid. If there's, well, water in this case is not charged, so there won't be a gradient in the electric potential, and there's, the concentration is in fact the density of the water. So in the case of water, its gradient of electrochemical potential will be essentially the gradient in the pressure. And that will be resisted by a frictional force between the solid matrix, which is porous, and the solvent. And this frictional coefficient is directly related to the Darcy permeability coefficient. So this first part of the equation is Darcy's law. Okay? And there's also, of course, if there's friction between the solute and the solvent, there will be an effect that comes from this friction coefficient, which also appears in the equation of motion for the solute. This is the equation of motion for the solute. The resistance as the solute diffuses through the solvent, that frictional resistance gives you Fick's first law of diffusion. You can recover the diffusivity from the inverse of this friction coefficient. So the only thing that we did differently is to go back to fundamentals and rederive these equations from scratch, and we discovered that a term that should not be neglected is the friction between the solute and the solid matrix. Because most people who do diffusion only do it in fluids. People haven't necessarily thought of including the friction coefficient between the solid and the solute. When you put that term in, you immediately discover that the diffusion coefficient inside the tissue should be less than outside of the tissue, which is a result that you expect. You also come to understand that just like the solid matrix can resist the motion of the solute, because the solute now collides with the solid matrix just like it collides with the solvent, but if the solid matrix itself is moving because it's being dynamically loaded, it can push the solute. Just like you have diffusion convection in fluids, where the convective motion of the solvent will drag the solute with it, it turns out that the convective motion of the solid will drive the solute with it as well. And so what we discovered is if you dynamically load these cylinders, this is the passive response, no dynamic loading. Here we found that theory predicted that we should be pumping the solute in because of this motion of the solid matrix going back and forth as you dynamically load. And this result was completely unexpected, never predicted in the literature. No one actually thought that this was something that should happen, at least in their papers. When you talk to people, they're like, isn't that intuitive? So I don't know. We'll see about that. So here's what the movie says. Oh, sorry. I got to start the movie. Okay, here we see, so you're dynamically loading the construct, and we're seeing that most of the effect starts out at the edge. You get a little spike in concentration. So we've already equilibrated the sample in a concentration where the concentration inside is equal to the bath concentration. That's this uniform value. As you dynamically load, you see that there's a spike that rises. This is actually a real-time simulation for essentially 1 minute's worth of dynamic loading. But of course, in the experiment, we're doing several hours of dynamic loading. So this gives you an idea of what the theory is predicting. A spike that progressively goes in. Eventually, this entire curve will rise up to give you a very large ratio of about 2 or 3 to 1. So we decided to validate this particular finding by running experiments. We used agarose gels, no chondrocytes. There's no tissue engineering here. This is just agarose gels and we used dextrans of different molecular weights to see the effect, because the theory predicted that the effect would be significant with larger molecular weight. The more the resistance between the solute and the gel, the greater this pumping effect would be. And here's what we found experimentally. Now, this is the passive diffusion control. There's no dynamic loading of the construct. This is looking at 7% agarose, 70 kilodalton dextran. And this is the concentration outside in the bath. So we've normalized. This is the concentration inside over the concentration outside. Because of steric volume exclusion, because not all the pores in the gel can contain a large molecule, under passive conditions, the actual concentration inside will be less than outside. That's why you get this value here is less than 1. With dynamic loading, we found this remarkable effect. First of all, we found a very clear effect of pumping, because this concentration is much higher than it would be under passive conditions. And after about 40 hours of loading, the concentration in the disc actually exceeded the concentration in the bath. So we even overcame the steric volume exclusion, meaning that those pores that are large enough now contain much, much more solute than the external bath. The pores that are too small to have any solutes still don't contain any solutes. The dynamic loading is not pushing any solutes in there. So we wanted to know if, since we're using these gels, are we perhaps damaging the gels with dynamic loading? Let me just remind you that the denser the gel is, the greater the resistance between the solute and the gel. So we found the greatest enhancement. So this is now the ratio between dynamic loading and passive conditions. You get a 15-fold enhancement with the higher concentration gel and the higher molecular weights. This is now 500 kilodalton. The effect is less significant for smaller molecular weights. And if you increase the gel density, you also increase the resistance between the solute and the gel, and you also see the highest enhancement with the denser gels for a given molecular weight. All of this is in agreement with the theory, which was very nice. Now let's take a look at whether we're doing anything special. Maybe we're damaging the gel with dynamic loading. Maybe we're enlarging the pores, and maybe that's the reason why we're getting more solutes in. So what does the theory predict if you stop loading? If you stop loading, the theory predicts that the solute concentration should go back to the passive limit. But if we damage the gel, we cracked it, or we produced larger pores due to perhaps plastic deformation, then it wouldn't go back down to the passive limit. So here's the experiment where we had finished loading for quite a while. This is the enhancement that we found over the passive limit, and this is the control under passive diffusion. And after we stop loading, if you wait long enough, the solute will diffuse back out, and these two values are not statistically different. So we confirm experimentally that we didn't do any permanent damage, nor do we have to worry about perhaps the solute binding to the gel, because it completely recovers after you stop loading. So it's a very interesting outcome that was predicted from theory and that had not been anticipated in the past, that dynamic loading can pump solutes in, and when you stop loading, they'll diffuse back out. I'm very proud of this discovery. It took us a long time, and it was resisted by a lot of people when we submitted grant proposals and all. So we had to piggyback these experiments on studies that were not funded. So anyway, so we further validated it by actually measuring the material properties that go into the model. I'll go over this a little bit quicker. So these are the properties that we need to measure. And these are the experiments that we conducted. We measured the tensile modulus experimentally. We measured the hydraulic permeability experimentally. We measured the diffusivity using FRAP experimentally for these gels. We plugged these properties back into the equations, and then we compared the predictions from the model to the experimental results. And what we found are, these are the predictions, and given that this is all experimental with a lot of uncertainty in the measurements, and that the fact that they were done on different gels at different times, it's remarkable how good the agreement is. Now we wanted to see whether we could see this enhancement in articular cartilage, because again, dextrans are not necessarily biological molecules, and articular cartilage may have some of these solutes bind to it. This is an immature cartilage construct. You actually clearly see those canals that I had mentioned to you earlier, which actually provide a pathway perhaps for the solutes to transport. Here's the mechanical device where we loaded these constructs, and here's what we found. passive conditions, no dynamic loading, the concentration equilibrates. This is again 70 kilodalton dextran. With dynamic loading, we find an enhancement very early on, and it keeps improving over time. Now, we were very curious why there was this sudden jump after only 30 seconds of loading. That kind of didn't go with the theory. The theory still predicted that you should rise from the same amount. And so we had to do confocal microscopy to understand that better. What we found is, this is the passive diffusion. Even though there is solute in the canals, it's not diffusing much because it takes a long time for the 70 kilodalton dextran to diffuse within the very dense cartilage. When you dynamically load, even after 30 seconds only, you can see high concentrations of solute right around the canals. So there's this immediate pumping effect that I had shown you in that little movie, those little spikes of concentrations that start out at the edge and over time start to propagate inward. That's exactly what you see, but not just around the outer periphery. At every canal, this phenomenon will occur. That's the reason why you get this very large enhancement early on. And over time, you can see it diffuses further and further into the extracellular matrix away from these canals. So what we have discovered then is not only that we can pump solutes from an external bath, we can pump solutes out of the vascularization. And so the implication goes beyond just articular cartilage, that perhaps this is the way the body works in tissues that undergo regular loading, like your heart muscle or your lungs, other tissues, tendons. It could be that this process of pumping large molecules out of your blood vessels is more pervasive than people realize. All we've done is shown it in this dead tissue here. These are not live cartilage explants, so we cannot, over-predict what's happening in the body. But to me, I feel like it's a fairly fundamental discovery that mechanical loading can pump solutes out of blood vessels. So it's a very interesting outcome. And we decided to do it with, again, different molecular weights. We find the same effect. If we only use fluorescein, which is the smallest molecular weight, there will be no enhancement due to dynamic loading. And if we use transferrin, okay, too much data. If we use transferrin, which is a molecule found in blood, which has a molecular weight of 70 kilodalton, we find the same enhancement. So this is a biological molecule that is found in blood that plays a role in binding iron ions, and which is important for collagen synthesis, and we find the same effect. So all of these enhancements occur not just for dextrans, and this recovery after you stop loading dynamically can also be found in articular cartilage, just like in the gels. So this phenomenon has now been confirmed in a couple of places. So we're quite confident that these results are real. Now comes the next question. Let's go back to articular cartilage and tissue engineering. So since we see an enhancement of solute transport, and earlier I told you you need more nutrients to make the tissue grow better, what I haven't told you is, what are those nutrients? And we don't know which nutrients in normal culture media are more critical than others. Part of our current grant is to selectively remove each of the nutrients to identify which ones are critical, and then do some modeling on how much dynamic loading might enhance the transport of those particular nutrients. But an obvious solute that people might consider are growth factors, and we decided to look at TGF-beta. So this is not completely thematically related to what I said before. It was for us when we first started this study. So we were wondering, if you can enhance the transport of nutrients from an external bath, since adult cartilage is avascular, it gets most of its nutrients from synovial fluid. That's what the literature says. So the question is, are there growth factors in synovial fluid? The literature says yes, there are. And what effect would growth factors such as TGF-beta have on articular cartilage? So the literature tells you that in general, TGF-beta's role is to inhibit cell proliferation and enhance extracellular matrix production. So that's true in general. This is not a review paper for cartilage in particular. What's interesting is that actually Barbara Boyan, who's one of your faculty members, has done some of the work on characterizing the role of TGF-beta specifically in articular cartilage. Teresa Morales in earlier studies showed that you can enhance the synthesis of proteoglycans by looking at the rate at which sulfate is incorporated into the tissue for different concentrations of growth factors. Let's look at these concentrations. We're looking at values that range up to about 20 nanograms per ml. And keep those concentrations in mind because I'm going to come back to this point a couple more times. So what you see is with increasing concentrations you get an improvement up to a certain point, at which point it plateaus, or perhaps you get an inhibitory response, so you may not want too much growth factor in your culture medium. This is a study that we did, just published at the OS recently, just out of curiosity to see in these immature bovine explants that we were testing, would we enhance collagen or proteoglycan synthesis? The answer was no. We didn't find an enhancement in either. What we found is that the tissue would swell less, it would degrade less with higher concentrations of TGF-beta. So we vary our concentrations in the range from 0 to 25 nanograms per ml. That's the swelling ratio. If you don't actually put any growth factor, the tissue will swell to twice its initial volume. So it swells a lot, which means something is degrading in it. When we measure the GAG content and the collagen content, it doesn't change. So the dominant matrix molecules are not changing. There must be minor matrix molecules, which we have yet to identify, which play an important role. Perhaps those exact same molecules that make the tissue stiffer when we dynamically load the tissue construct stiffer. So we wanted to know, what do we need to know about growth factors? So unlike transferrin or dextrans, growth factors do bind to the extracellular matrix. It turns out that TGF-beta is a fairly complex protein. What happens is this is the actual TGF-beta peptide. It's a dimer. It's 25 kilodaltons, so it's pretty large. But when the cells synthesize the TGF-beta, they synthesize it in what we call this large complex. This latent TGF-beta binding protein binds to this latency-associated peptide that you see in this shape, which itself binds to the TGF-beta. So it is well known that you must activate the TGF-beta. You must have this molecule separate from these other molecules in order for the TGF-beta to have an effect. When you purchase TGF-beta from Sigma, it comes in its active form. But this is what the cells produce. Okay? So the question is, what would activate the TGF-beta that is produced naturally in synovial fluid? The literature says that in general, in many tissues, the activation of TGF-beta comes through proteases like plasmins and some MMPs and reactive oxygen species. Don't ask me too many questions on this. Remember, I'm a mechanical engineer. But we have to read the literature and figure these things out. What was very interesting is very recently it was found that you can actually have integrin-mediated activation, meaning if you pull on these integrins that are actually bound to the large complex, you will free the TGF-beta peptide, which will then bind to a receptor perhaps on the same cell. So this is the first evidence of a mechanical action, a mechanical role to activate TGF-beta. Another very interesting study, which is also very recent, is that the shearing of serum, so this is mechanical shearing of serum, will actually activate TGF-beta. Let's take a look. This graph has two axes. The pinkish axis shows you the total amount of TGF-beta-1 in nanograms per ml available in serum. Now remember, serum has the content of cells that have lysed. So the platelets have released most of this growth factor. This growth factor is not normally present in the plasma unless you actually have serum, and the concentrations are on the order of 80 nanograms per ml, significantly higher than the concentrations needed to induce an active response, if you want, of TGF-beta on cartilage, according to the Morales study. If you shear the serum, then you will, over time, about 3 hours here, 4 hours, I can't count, you will actually activate about 4 to 5 nanograms per ml. Of these 80 nanograms will be activated, which is a very interesting result. Again, another evidence that mechanical effects, in this case shearing of serum, will activate TGF-beta. So we asked ourselves, could the shearing of synovial fluid activate the TGF-beta that's present in synovial fluid? And so here's what synovial fluid looks like. It's kind of this yellowish viscous liquid. The first thing that we did was to characterize the total amount of TGF-beta in synovial fluid it's on the order of 5 nanograms per ml. Not as high as in serum, which has about 80 nanograms per ml. Of this TGF-beta, a negligible fraction is normally active, meaning that most of it is an inactive form in synovial fluid. So we asked the question, if we shear synovial fluid using physiological shear rates, would we activate the growth factor? And here's what we found. This is the amount of time that we're shearing it. Here's the control where the synovial fluid is not being sheared, and these are different shear rates, all of which are actually subphysiological. It turns out that synovial fluid shear rates are very high in your joints because you have sliding velocities on the order of, say, 10 millimeters per second, and film thickness on the order of 0.1 micron. So we're talking about shear rates on the order of 10 to the 4 reciprocal seconds. So we're here like one order of magnitude less than that. And within less than 8 hours, we have activated about 1 nanogram per ml out of the 5 nanograms per ml that we have. So this is, again, a very interesting discovery. For musculoskeletal systems, this is the first evidence that I can see in the literature of a mechanical effect for activating a growth factor. And so this growth factor is presumably now available to diffuse into the cartilage, right? Because the synovial fluid coats the articular cartilage. So then the next question is, okay, how much growth factor is there in articular cartilage to begin with? So let's take a look at that. Again, the literature has, for those of you who have a chemical engineering background, is this activation reversible? Meaning if this active TGF-beta peptide is sitting in a synovial fluid where the LOP, the small complex, is still available, the large complex is still available, will they bind back together? The answer is no. Over an additional 8 hours, we find that the concentration of active TGF-beta remains constant. That's an important result. So it turns out it's a very stable form. Active TGF-beta, remember there are no cells to consume or use these growth factors in this study, so therefore the concentration stays high. Now, here's the concentration of TGF-beta in cartilage. There's only like a couple of studies that have measured it before, and one of the studies said that there would be concentrations of up to about 200 nanograms per ml, and we couldn't believe that that number was real. That's a huge amount. Remember, in synovial fluid, it's about 5 nanogram per ml, and we actually confirmed that you can actually have just a huge amount of growth factor that is already in the cartilage extracellular matrix. But if you expose the cartilage to a bath that contains 1.5 nanograms per ml of TGF-beta, after about 4 hours, the concentration almost doubles. That means that there are a huge number of binding sites in cartilage for this growth factor to stick to. So you have a binding effect. It goes in, it binds, so it's no longer in soluble form. So as far as the tissue is concerned, the concentration of soluble growth factor is 0. So more will keep diffusing in until you fill up all these binding sites. And it looks like there's plenty more. This is only after 4 hours. We were running out of TGF-beta in a bath. It was actually the concentration in the bath was going down during these experiments. And if you put 6.5 nanograms per ml, you can keep getting more and more TGF-beta into the tissue. So what you realize is cartilage not only starts out with a lot of proteoglycans, but you can get a lot more in. But what's important to know is this is actually the active form. And the fact that the active form is binding as well is an interesting finding. So it turns out that not only the latent form, but also the active form of TGF-beta will stick like crazy to the extracellular matrix. What does the tissue do with all this TGF-beta? We're not totally sure, since those experiments I showed you earlier don't show such an obvious effect. Why would the tissue need to have such large amounts of growth factor is a good question. The literature says to decrease cell proliferation and cellularity of articular cartilage is very low. In immature cartilage, only 10% of the tissue volume is cells. So perhaps that's the main role of the growth factor, is to make sure that the cells don't proliferate. And you just, you have plenty of TGF-beta stored in there to help you maintain that low cell content. We don't know. Anyway, this is where we are now, and we still want to look at the effect of dynamic loading. So we looked at the effect of shearing synovial fluid to activate the TGF-beta. Now we want to see the effect of dynamic loading and whether it enhances the uptake of TGF-beta further. So I can give you some concluding remarks. We looked at solute transport, and we looked at TGF-beta as perhaps an interesting solute to analyze, but the level of complexity with TGF-beta rises exponentially compared to dextrans or transferrins, so we have a lot more work to do. But in the end, it's still geared towards understanding better what's going to allow us to engineer cartilage better. So in the end, we're still hoping that we're going to create these full-size patella constructs which are actually functional, that can be implanted in the body. So I'd like to, of course, acknowledge these funding agencies, well, these grants from the National Institutes of Health. Mike Albro, who was the student who did a lot of the solute transport studies, now is doing a postdoc with me with the TGF-beta. Clark Hung, that I mentioned earlier, is my very close collaborator on these tissue engineering studies that we did. You know, most of the experimental studies of tissue engineering were done in his lab. Jimmy Cook is a veterinarian with whom we collaborate, where we're now trying to implant some of these constructs into animals to find out how effective they are. And I'd like to thank you for your attention.
Okay. Time for questions.
A few questions. One is that, are these on gels without cells or with cells? Then what is causing the asymmetry of that it sucks in and not, because we go both ways?
So
microfracture is the third thing. Do they put in channels that way too, essentially?
Okay, so lots of questions. The solute uptake studies with the dextran don't contain chondrocytes. The reason the process is asymmetric has to do with the balance of the solvent properties in the gel and the solute properties. The simplest way to understand it is that first compression, that first one that gives you that spike. If you compress the gel and let's say the solvent leaves faster than the solute, usually the solvent will only leave at the periphery. It takes a long time to leave from the center. So you'll get this effect of concentration right at the edge, this spike in the concentration. Then when you unload, the matrix recoils, pushes that spike inward. That's the simple asymmetry in the mechanism. So I'm skipping all the details of the theory, but the properties have to be in the right ratio to give you this enhancement. But in the end, it comes down to, in practice, as to the molecular weight of the solute.
Yeah, because if you take it out for a long period, then it all is reversible.
Yes, exactly. It is all reversible. Therefore, that's why we don't think, that there are any fundamental alterations to the gel. Now, microfracture in orthopedics, you mean the procedure where people actually fracture the bone? The main reason for that is to bring in the blood supply. The surgeons realize that in order to grow any tissue, you've got to bring in the bone marrow and the blood supply. So that's why they cause these microfractures to the bone. And it grows a fibrocartilaginous tissue that doesn't have the same properties as articular cartilage.
Putting channels in.
Well, I mean, they're definitely putting channels in temporarily, but yes, whether it's pumping things out, it could. But remember, there's no soft tissue layer on top to do the pumping. It's just bone with holes in it. Yep.
Gerard, great talk as always.
Thank you.
So going back to the design of the construct.
Yep.
And, your theory predicts a big effect on the permeability of the tissue. So the way you guys explored it experimentally was one big hole. What about many little holes instead? Would that also, I understand that it depends on the molecular weight of your solute, but I would imagine that would be potentially even better. We have many little holes than one fat big hole.
Right. So we haven't varied the size of the hole. We went with what was easy to perform. I guess you could just puncture with a needle to get a smaller hole and just do many of them. What Clark has done in his studies, which is interesting, is he's also looked at other alternatives, such as creating microbubbles in the gel during the process of gelling the constructs. And you can use ultrasounds or other methods. Actually, Dr. Bellamconda has developed a technique that Clark adopted for creating, supplies of, nutrients as well inside the gel. And they actually show better results too. So you can create essentially microcavities about 2 microns in diameter, which, will store some nutrients temporarily, and then the tissue constructs after 4 weeks have better properties. So there's no reason to doubt the idea that smaller channels might do just as well. So we might do that. There's only so many parameters we've varied so far, but we could try.
The surgeons may not see the little hole.
Yeah, that's right. They won't complain as much. But they will fill up faster. So the question is whether the effect will last long enough.
Jen?
So 2 questions. One is follow-up of what David asked. I can understand the pumping effect too, because you started out inside the tissue low. Right, so that I can understand that you get them in by mechanical pumping. What I don't still trying to understand is how do you get it higher than outside?
Yeah. So actually, the movie of the theory pumping in, I decided purposefully to start with the initial condition that the concentration inside is not 0, it's equilibrated with the outside, just to show that the theory is actually able to predict this. But from a simple perspective, people are familiar with the concept of pumping solutes across a membrane, right? It's just that the pumping mechanism there is specifically tied to a molecular motor that changes its conformation, will take one ion from outside, pump it in, or vice versa. So the idea that you can pump solutes and maintain a gradient in concentration across a particular boundary is not foreign to people who understand cell physiology. What we've simply discovered is this mechanism isn't limited to molecular motors in membranes. So you're providing mechanical energy. So you're adding energy to the system. This isn't a zero energy system. You're pumping. Just like a pump can pump water up a gradient, just like you can pump water from the bottom of a building to the roof, you should be able to pump solutes. There's nothing to prevent you from doing that. What we're saying is, this mechanism shows that it occurs at the boundary with you're pushing on the solute in an asymmetric fashion. You're going back and forth. The amount that you're pushing out is less than the amount you're pushing in. It is not something you could have guessed intuitively, and I understand that perhaps it's not as... Well, thermodynamically, it's very easy to realize that there is an energy input, and that's the important thing. So it is a form of active transport. So when we first published these results, we put the term active transport in the title. It irritated so many people that I stopped using the term. Some people are rubbed the wrong way, so I'm not going to use it. But active transport fundamentally means you have an energy supply, from ATP that's activating your molecular motor. Here, the energy supply is the device that's mechanically loading the tissue, or in your body, your own joints.
Right. Let me ask the second question, which has to do with TGF-beta.
Yep.
So was the active form and inactive form in a native state can be converted reversibly, or it can only go one way? In your test, you convert it into going one way.
Right. Well, we didn't. We activated it, and we waited to see whether it would bind back to the large complex, and it didn't. And from what we've read in the literature, usually when it's been activated in the body, either the active peptide binds itself to the matrix and it now is available to be used by the cell if it's close enough to the cell, or it is taken up by the cell, and we've read that within, I think, 30 seconds it's internalized, and or something on the order of 30 seconds it's internalized by the cell. So the cell can either break down the growth factor, or the more intriguing outcome is apparently, and that I have to see more papers to confirm it, you can have active TGF-beta that is not taken up by the cells and yet is able to influence the cell function.
So I guess what I was getting at is what assay do you use to determine its active form? I mean, use an antibody that recognizes the active epitope, or it's actually testing its functionality of stimulating tissue growth?
So what we have is we first assay it. We have an antibody that doesn't distinguish between the active and inactive form. We got this from Sigma. And then, so we assay the content, then we activate it using a lower pH, which is a well-recognized method for activating TGF-beta. You can do that in vitro without any problem. And then we compare the amount from the 2 measurements before and after pH activation to figure out how much is in the active form, how much was in the inactive form. That's how we've been doing it.
Does the mechanical loading claim that the matrix also activate latent TGF-beta?
That is-
It's bound in the matrix.
That is one of the experiments we've started doing, and so far we haven't been. So one of the things that was holding us back was we didn't know how much TGF-beta was in the tissue. So now you see there's a huge amount. So if it activates, but it only activates, say, 1 nanogram per ml, it's probably going to be noise behind the 200 nanograms per ml that's already present there. So far in our preliminary studies, we haven't seen an obvious activation. But I do suspect it's there. It's just this one is not a low-hanging fruit. It's a harder one to figure out.
Any other questions? I have one
Just
more
a quick question. I think this is sort of along the lines of what Andres was asking. I guess another approach, instead of creating the whole construct with holes, might be like a layer by layer type of thing, where you, over time, construct it so that you know, maybe you have a thin layer and it gets the properties. And then I guess what I was wondering is, if you added another layer, would it adhere to the initial layer? Or have you thought about that?
So in the field of cartilage tissue engineering, this idea of integrating, you know, say the construct with the surrounding tissue is a big deal. Bob Saw at UCSD has done a lot of work. The bottom line is it's very difficult to adhere two pieces that you've engineered yourself and get the same bond as if you had grown it from scratch as a whole. So it would be a greater challenge to do that than perhaps what we're doing now. And we've tried also to see what is the adhesion, you know, potential of our tissue constructs, and it's very low unless you do something special. You have to degrade proteoglycans, you might have to degrade the collagen. It's the collagen matrix that's the biggest challenge. It doesn't seem to travel across an interface and, you know, connect with collagen produced on the other side.
All right. Well, let's thank Gerard again for a wonderful